Dr. John Speakman, welcome to the show.
Hi, thanks for inviting me.
Well, I've had a lot of your colleagues on, and rivals, you could say, although I don't know that we have rivals in science. But I'm excited to speak with you. It's been a long time coming here. So you're a biologist at both the University of Aberdeen and the Chinese Academy of Sciences in Shenzhen. So you're going back and forth between the UK and China. And
yeah, so initially I was traveling a lot more than I do now. So I was trying to do it on a kind of 6 weeks here, 2 weeks in the UK schedule, but that meant I was jet lagged for 4 weeks every sort of 2 months, so I just go back in the summer now to the UK.
Wow, that's a rough time difference. So anyways, you are one of the world's foremost experts on energy balance, so we're gonna talk all about
Thank
that.
you.
And to get started, there's something called the doubly labeled water method. Okay, so this is obviously what your bread and butter is, so to speak. You spent decades applying and refining this approach, which is a way to measure energy expenditure. So many people probably aren't familiar with this, but it's kind of essential for this discussion. So can you explain the technique and why is it such a game changer in the field?
Yeah, so I'll start with why it's a game changer, because it's the only technique that we have that can measure your energy expenditure when you're out there free living, when you're doing all the things that you do as a human being. It can pick up all those expenditures of energy and give you a sort of integrated total over a period of about a week to 2 weeks. So there's no other method gives us that answer, and that answer is pretty important in lots of different areas. So having this technique available makes a big difference in our understanding in lots of areas.
Okay.
So how it works, if you think about your body is made mostly of water, and there's a constant flow of water coming into your body as you drink, as you eat food, and also metabolic water produced from our metabolism. And then there's also constant losses of water in our feces and urine and evaporation. So if I was to take some water with isotopes of oxygen and hydrogen in it and give it to you to drink, it would spread through your body and it would then get washed out in all these elimination routes. So the losses of hydrogen and oxygen would be kind of roughly the same because of that sort of flow of water through your body. However, oxygen has another route because we constantly consume oxygen and we exhale carbon dioxide. So when we exhale carbon dioxide, that also takes with it some of the oxygen. That's not a completely intuitive thing because, of course, oxygen molecules bound up to carbon in carbon dioxide, and we're putting in oxygen in water. It's not entirely obvious how those oxygen molecules get from the water onto the CO2. And actually, it's because we often think about, the water in our body is just water, but actually it's constantly being exchanged with other compounds in our bodies. And there's a reaction with carbon dioxide via bicarbonate that's catalyzed by an enzyme called carbonic anhydrase. And that swaps the oxygen back and forwards between CO2 molecules and water molecules. And so if I put oxygen into your body in water, and oxygen 18 into your body in water, it will get into the CO2 molecules and those molecules will be exhaled. And so there's a divergence in the reduction of the two isotopes. So the hydrogen comes down over time. Actually, it's an exponential decline because only of the water flux, whereas the oxygen comes down because of the water flux and the CO2 flux. And therefore, from the difference in the elimination curves, we can tell the CO2 production, and the CO2 production is related to the energy expenditure. So it's a pretty cool idea. It was the idea for it actually came about in the 1950s. So there was a guy called Nathan Lifson working at the University of Minnesota, and he came up with this idea. He was the first person who showed this equilibration of the oxygen isotope. And he came up with this idea, and he did a few studies in mice in the late 1950s. And he published a paper in 1975 saying that the main problem with this method is it's super expensive because the oxygen 18 isotope is pretty expensive to produce. And so in this 1975 paper, he said that this technique is really nice, but it will never, ever be used in humans just because it's too expensive. And he estimated it would cost about $15,000 per person to apply the method. So it was a surprise then that 1982, first measurements of humans were made by Dale Scholler, who was then at the University of Chicago. And the reason that it became possible was because of improvements in the technology. So it became possible for us to detect lower and lower levels of these isotopes. And because we could do that, we had to put in less. And so the cost of the technique declined. And the current cost is around about sort of $600 to $1,000 per measurement. So it's still not cheap, but it's much cheaper than it was projected to be in 1975. And now it's used probably, I would guess, has been used in total, we estimate around about 30 to 40,000 people. So there's lots of measurements out there that people have made. So I should add one thing is that people are probably thinking isotopes, it's dangerous, it's, radioactivity. So these isotopes that we put in the body are deuterium and oxygen-18. They're both stable isotopes, and they're actually already in your body. So your body has about 155 ppm deuterium and about 2,000 ppm oxygen-18. So they're already there, and all we're doing is tweaking the values a little bit. So we push up the deuterium to about 300 and the oxygen up to about 2,200. And then we look at it going back down to the background level. So it's completely safe. It's used on pregnant women and premature babies, and there's no evidence of any side effects whatsoever of it.
Okay. So a lot of technical details, but essentially it lets you measure energy expenditure in a very accurate way. It's a gold standard. It's kind of what other attempts and metrics are sort of compared to. And so I had Herman Poncer on previously, and you were one of the senior authors on the same paper that he was a lead author on in Science in 2021. So that kind of we talked about how it mapped energy expenditure across a lifespan from, infancy to older adulthood. So just before we get into more details, did you have any sort of most surprising finding based on your knowledge of energy expenditure before that study was done?
I mean, I think I had ideas about how it might play out, but not based on evidence. I think the beauty of that paper was we pulled together data from lots of different sources into this database, and that allowed us to look across the lifespan, which had never been done previously. And there were some big surprises there. So, a priori, if you just looked at when would you think the big changes in your life would happen? Everybody would kind of say, adolescence, you're going through lots of sort of rearrangements there. You're sleeping half the day. And then, so there's probably going to be some effect on your energy expenditure then. And then middle age, that's when your sort of gut starts to expand. And, everybody thinks there's a kind of midlife reduction in your metabolism. Turns out both of those things were completely wrong. So puberty, not a blip on the graph at all. Middle age, no sign of any reduction whatsoever. So your metabolism seems to be constant from your late 20s through to about 65 or so. It's just completely flat. There's no evidence that there's any reduction there.
So I think that's super surprising for people to hear. And I'm also curious on your explanation for why is that the case? And I think one thing that we probably need to discuss, I'd like to hear your thoughts on this, is when you, in general, if we think of wealthy populations like in the US or the UK, Australia, high-income countries, people as they get older tend to lose lean mass and gain weight. And in general, there's a loss of this muscle mass and an increase in weight. So is that part of the story here? Like, is that explaining maybe some of this that's going on? Like, because people do gain weight, are they actually? Is there energy expenditure? Are you adjusting for lean mass, or how does that work exactly?
Yeah, so this is all adjusted. So if you adjust for the level, obviously the more lean tissue you have, the higher your energy expenditure. So we have to take that out of the equation. So when you remove that from the equation, it's still completely flat through these mid-years. So it's not because there's some adjustment happening in the size of the fat-free mass. In fact, fat-free mass doesn't start to decline until you get pretty old. And actually, and it's one thing we should sort of really note in these data is it's all cross-sectional data. So actually, we found that there's a reduction in metabolic rate after the age of about 60, 65 or so. It starts to go down, or at least it looks like it starts to go down when you look at the graph, but these are all different individuals. So we actually don't know whether it really goes down, because it could be a sort of survivorship bias. So what could happen is the people with the high metabolic rates might die, and all you're left with is people with lower metabolic rates. So it looks like your metabolic rate's going down as you get older, but that may not be true. We're not actually clear on that yet.
So, okay, so if I understand correctly, even when you adjust, so you're basically accounting for, you're holding lean mass constant, you're holding BMI constant, or your body weight constant. And the metabolic rate
just the fat mass and fat-free mass.
Fat mass and fat-free mass. Okay, so you're holding those constant, and it's still, metabolic rate stays completely the same until you're about late 50s, maybe early 60s, right?
Yeah.
Okay.
From mid-20s. Okay, so
from mid-20s.
Yeah.
Yeah, I think it's just fascinating. People think about and it's somewhat true. Like when you're in high school, when you're an adolescent, you can eat a lot more food and not gain weight because your energy expenditure is high, but it does taper off right around those college years. And I think what people sort of assume is that the metabolism slows down in their 30s and 40s and 50s, and it doesn't really happen until your end of your 50s and 60s. So, okay, now you've also been, you've been kind of one of the prominent figures here in understanding the obesity epidemic. You've described it as a pandemic. So a big question in the field, is whether the main driver is on the intake side, how many calories are we consuming, and the expenditure side. And so when you've looked at this over decades, where do you come down on that debate?
Yeah, so that's a super interesting question, and obviously there's a lot of kind of anecdotal evidence there that our lifestyles have changed a lot since 1950s through time. So, if you look back to 1950s and 1960s, the types of things people were doing then were more active jobs. There were less car ownership. People used to go down the shops with shopping bags and carry them home, and so there's a lot of like anecdotal evidence that our physical activity has changed over time compared to what we're doing now. And I was pretty interested in trying to quantify what the reduction in energy expenditure had been, so we could get a handle on that. We could say, okay, not only has energy expenditure gone down, but it's gone down by this number of calories. And the way to do that would have been if somebody in the 1950s had thought there's going to be an obesity pandemic, why don't we measure a group of people every year and then we can see what the decline has been. So there's two problems with that. First, of course, nobody predicted there was going to be an obesity pandemic, but also the technique wasn't available until the 1980s. So what happened in the early 2000s was I kind of looked around, and there's this guy in the Netherlands called Klaas Westerterp, and he was one of the first people who used the method, the doubly labeled water method. So his first measurements were in 1985. So just, 3 years after the very first application of it. And I went to him and said, look, you've done lots of studies through time here. why don't I come to your lab and pull together all the control groups, because they're effectively a sort of random sample through time, and we can see what this decline has been in the control groups. So he said, yeah, come over. So I went over to Maastricht in the Netherlands and I pulled out the data from his papers, and it turned out there's nothing. It's completely flat. So this seemed like there was no reduction in energy expenditure at all. And we published that in International Journal of Obesity in 2007. And there were lots of people who said, yeah, but there's big problems with that, because actually in the Netherlands, there wasn't really an obesity epidemic going on. So you've measured, energy expenditure through time at a time when there wasn't an obesity epidemic. So what? So anyway, much later, once we've put together this big database that had much, much more data in it. So the paper with class had about 300 data points in it. So now we've got about 6,000 data points. And they're also like 80% of them are from the US. So nobody could then say, you don't have, a sort of the right area and the right time frame to do this. And so I pulled together all that data and we looked at it, and sure enough, when you look in that bigger sample, there's a reduction. So the total energy expenditure has come down over time, and I thought, great, that's really good. Now, that doesn't necessarily mean there's been a reduction in physical activity causing that, but you can get a handle on that because we can also measure the resting metabolic rate. And that had been done in about 1,500, 1,800 of these 6,000 people. And so what you can do is you can do it two ways. You can either calculate the difference between the basal metabolism and the total, and that gives you the activity energy expenditure, or you can calculate the ratio of the two. So it doesn't really matter which one you do, because the answer comes out kind of the same. So when we did that, if you look at what happens to the physical activity energy expenditure, it doesn't change. So it's flat through time. And that can only come about through the basal metabolism going down. So if the total's going down and the activity is constant, then it means the basal is coming down. And nobody had ever really thought about that as a potential factor, because everybody just said, okay, well, basal metabolism to basal metabolism, it's going to be flat, So that was a real surprise that basal metabolism seems to have declined. And it was such a surprise that we didn't really believe it. And so we went back to the literature and we pulled all the data that we could find on basal metabolism, going back to the first measures by Harris and Benedict in the 1920s. And sure enough, there's been a decline over time. Over that whole time period since the 1920s, there's been this steady decline in basal metabolism, which is actually mirrored by a very small decline in our body temperature as well. So that is a potential component of making us more susceptible to obesity. So we can look at how much it's changed, and we can do a bit of modeling, and it's there, but it's not enough. And so it's clear that even if there is a component that's attached to this BMR, the main driver is increasing food intake. So there is a small effect of changing metabolism. It's not due to changes in physical activity, despite all this anecdotal data. It's due to this very small change in our basal metabolic rate.
Okay, let's talk about this. But first, why is it, and maybe you don't have the answer, but why is it that the basal metabolic rate has been declining for the last 100 years?
So we don't know. It's basically the answer, but we have a few ideas. So you had Herman on recently. Herman's thought is that it's principally because we don't spend as much energy on immune function nowadays compared to 1920s, 1930s. And that's possible. I mean, obviously, there's been lots of vaccine campaigns, lots of changes in our immune environment. So we're much less susceptible to infectious diseases. And so we don't need to run a really powerful immune system anymore. So that's potentially there. There's no evidence to support it, but it's a good idea. Another idea is there's been some big changes in our diet. And so if you look particularly at the fats that we eat, so if you go back to the 1910s, 1915s, almost all the fat that we ate was saturated fat, butter, tallow from beef products, those sorts of things. And that over time has steadily declined. And from the 1950s, we've had a big expansion in seed oils in our diet. So linoleic acid in particular has increased enormously through time. And of course, there's a big seed oil, group who thinks seed oils cause everything. But one thing we had is some data from mice that have been fed different fatty acid diets. And it seemed that linoleic acid was associated with a lower basal metabolism in those animals. So that's another potential driver of the reduction in basal metabolic rate. So the problem with that study was it wasn't completely controlled. So there were some covariances and it's a statistical association. So what we've done since, and this is unpublished, is a much more refined experiment where we've fixed the monounsaturated fatty acids at a given level. We've fixed the total fat at a certain level. And then we've just traded off saturated fat against unsaturated fat. And when we do that, we get the same result. So there is a reduction in basal metabolic rate that's attached to the increase in linoleic acid in our diet.
Interesting. So if I'm hearing you correctly, there's a couple different reasons for the drop in the basal metabolic rate. One is potential is the potential reduction in our immune system needing, we don't have as much infection burden. We have vaccines. And then also the diet shifted from a lot of these saturated fats, animal fats like butter and tallow, to these polyunsaturated fats in these industrial seed oils, right? So this is a small change. So I think we also need to caveat that the basal metabolic rate is probably a small part of the picture. Can we talk a little bit about the bigger piece of the picture, which is the actual foods that we're eating, right? We're eating very large excesses in calories, right, and energy. And so you've also done quite a bit of mouse studies where you look at different macronutrient compositions in the diet and body weight and stuff. Can you describe your findings there and what you think about maybe the roles of diet, in particular, maybe macronutrient combinations?
Yeah, sure. So there's been a lot of small studies in mice that have looked at this diet and that diet, and we wanted to really do a big study where we very systematically changed the diet. So what we did was we designed a kind of matrix of 30 different diets that changed in the protein content, carbohydrate content, and fat content. And the way that we set it up was such that you could unequivocally pull out the effects of these things. So there was, we got rid of the covariances with the design of the diet, because the problem with diet studies is they're not easy to do, because if, let's say you have this idea that reducing protein in the diet is important for driving your appetite. The question is, what do you replace it with? So if you reduce the protein in the diet, what do you put in there? Because whatever you put in there may be the thing that's actually causing the problem, not the reduced protein. And so it's quite difficult to design these diets. You have to have a sort of multitude of different manipulations going on. So we did that. We designed all these diets and we got a company to make them, and they were very well controlled in terms of the composition of the fats and things like that. And we did this study on 1,200 mice or so. So it was a big study. And there were a couple of big outcomes from that. So the first thing that we tested was this protein leverage idea. So the idea is from Steve Simpson and David Robenheimer. And basically what they suggest is that the thing that drives us to overconsume is what's not there rather than what is there. So basically what they've suggested is that through time, the protein content of our diet has declined. And so if we look at cheap ultra-processed food, for example, very low protein contents. And what they suggest is that we actually consume food not for its energy, we consume it for its protein. And so if you reduce the amount of protein, you have to eat more food to get your protein requirements. And then you overconsume calories. And so it's the low protein that drives us to overconsume calories. A really cool idea. And so we tested that in the mice. It doesn't work. So in the mice, there's no indication whatsoever that reducing protein from like 25% down to 5% has any effect whatsoever. So this is five different strains of mice exposed to these different diets. So protein leverage, if you're a mouse anyway, doesn't seem to be an important part of the picture. So then we come down to the big two, of course. So there's now a massive debate going on for the last sort of 30 years about whether it's carbohydrates or whether it's fat. And so what we found was a pretty interesting pattern as you change the fat content and the carbohydrate content, keeping protein constant. So what happens is, as you increase the fat content of the diet, the mice get fatter and fatter and fatter. But then they level off. So between about 40% and 60% fat in the diet by calories, it's pretty flat. And then if you go over that hill, it starts to go down again. And after that study, we followed up with another 6 diets that take it right down. And so what happens is there's a kind of mountain that you can imagine that on one side is low fat, on the other side is low carb. And you can think about these communities who believe in a sort of low fat is the thing, or low carb is the thing. They're like villages on either side of a mountain. Okay. Mm-hmm. And the people on the low carb side of the mountain, they tend to be people who've never been obese. And what they observe is that if they increase the amount of fat in their diet, they start to get fat. And so the sort of idea there is, okay, it must be fat. Fat's the thing that causes this problem. But the assumption is that if you eat more and more and more fat, it's just going to go up and up and up and up. And that's not true. Whereas on the other side of the mountain, you got the low carb people, and they're generally people who have been obese. And what they've noticed is as they cut carbs from their diet, they lose weight. And so they then have come down the mountain on the other side, and their view of the world is it's all due to the carbs. And their assumption is that if you go back up the mountain, it's just going to go up and up and up the other way. So in fact, both of them are wrong, because it's a peak. And it's a peak of around about this, composition where you have about 15 to 20% protein, you have about 40 to 50% fat, and the balance is carbohydrates, about 20 to 30% carbohydrates. And that, in a mouse, is the thing that makes a mouse fat, that sort of composition of food. So the big question is, is any of this relevant to humans? I mean, it took me a long part of my career to work out nobody cares if you find out why mice get fat. You've got to know why humans get fat. And the problem with measuring what humans eat is humans are not particularly good at reporting their food intake. And so it's very difficult to from epidemiological side, it's very difficult to tell exactly what people have been eating. And it's also super difficult to do food manipulation experiments over an appropriate period of time. So you really need to, lock people away for 6 months to a year and feed them a given food before you get a good idea about what's actually happening. But there's a guy called Ted Naiman and a guy called Anders Ehrenfeld who've set up this thing called satiety per calorie, and they get people to log their intake online. And I was very lucky to sort of speak with them, and they shared 90,000 data points from people who have logged what their food intake is. And I analyzed that data. So there's a lot of variation in there. So you've got to do quite a lot of screening. And I reduced it down to about 30,000 data points that we think are good data. And that peak is there. in that data. So if you look at the amount of calories that are consumed against the fat and carbohydrate content, it's got this same peak. And that peak's around about the same position that it is in the mice. So that sort of says to me that probably the mouse data is good. And probably what's happening in humans is a very similar thing, that, if you go low carb, it works. Nobody's questioning whether that works or not. If you stay low fat, it works. What's really bad is eating that combination. And that combination turns out to be all the things that we like to eat. Okay, so all the things like, cookies, chips, everything that's pizza, everything that's kind of bad sits on that mountain. And, that's why people get fat. They eat that food, and we think that it provides a reward signal in the brain. We have some evidence for that now. And it somehow circumvents your homeostatic intake mechanisms, and you overconsume calories. And, you go back to that food because it's nice to eat, and you overconsume calories, and before you know where you are, you're in a fat storage issue. And that's what I think is going on. So it's not fat, it's not carbohydrate, it's the mix that's the important thing.
Okay. So a lot to unpack there. So to start off, the mouse data, I think it's interesting that the protein leverage hypothesis you said didn't really pan out. However, I was talking with Christopher Gardner at Stanford just a couple of weeks ago, and he made kind of the point that in general, just about every population consumes between 15 to 20% protein, right? Like whether they're more plant-based or more animal-based, when they have a choice. Now, some of the populations in lower income contexts, and they have really nutrient poor, just cereal dominated or staple dominated, starchy staples, they may be on the low end. But when you have the preference, we see populations. So I don't think, when I think about this kind of from a rational common sense standpoint, I don't think you're going to get humans to eat 5% of obviously we have a minimum protein threshold, like we do. But what I hear, the way I interpret it is that you don't need just more and more and more protein. What the challenge really, what happens is when you combine those starches and those fats, which when you look at nature, I think breast milk may be the only food that's high in starch or sugar, carbohydrates, and fat, right? But most foods are high in one or the other. And so there's that combination. And I think the packaging and the processing, all of that also factors into it. But we've sort of created these foods that are highly palatable and we overeat, right? So I think that's one big piece of this puzzle. For the listener or the viewer, I had Andres Enfield on the show as well. He was the second guest. So we talked about the satiety per calorie. And, the other thing is that Ted Naiman as well, they really emphasize protein. So I'd be interested to know your take on their sort of strong emphasis on protein for satiety. Is that because that sort of by default keeps the carbohydrate and the fat lower in general, so you don't go too high in either? What is your take on the protein?
Yeah, I think that's interesting. In that data that they have from the Harbor app, there is a protein signal. So it seems that there is an effect of protein in humans that we don't pick up in the mice, and that's maybe a human mouse difference, but I don't think it's a big effect. I don't think it is bigger than the fat carb effect, and that makes. And you raise a really interesting point. Is okay, it's fine to say, well, it's rewarding for us to eat that, but an evolutionary question is, why is it rewarding? What foods have made it advantageous for us to have that in our brains? And actually, there are no foods. If you look, there's a few. It's like red oak or something like that. It's red oak seeds is something that has this combination. But there's no way
maybe acorns.
There's no way the entire human race has got fat because of red oak seeds. And so the thing is, it's got to be something that sets our brains up to do it. And you hit the nail on the head. We suggested a while back it's breast milk. So the composition of breast milk is exactly in that zone. And you can imagine why we would have a reward system in our brains that said, this is good stuff, and you need to eat it, because at that stage, that's your only source of food. So you have to learn that it's good, and you have a reward system in your brain. So the problem is it's never switched off. And the reason it's never switched off is because that never used to be a problem, because there's no foods in the environment that match that. So we never used to overconsume. The question is, why do we have that reward system in our brains? And if you look at the environment, I mean, it could be, there's a food in the environment that's good for us, and therefore we have a reward system. But there's nothing. If you look in the environment, there are no foods that match that exactly, except for some really odd things like red oak seeds. And so it's got to be something different than that. It's got to be something that we've all been set up to eat. And you said it yourself, the key thing is breast milk. So breast milk has that exact composition that makes us fat. And so you could imagine that what happened is, early in our lives, we need that system because we need to know what food is, and we need it to be rewarding so that we drink lots of it and we grow up. The problem is that system's never switched off, and it's not switched off because historically there was no need to switch it off. And so it just sits there in our brain. Come to modern society, and food companies, I mean, I don't think food companies are bad people. They just doing business, and they find things that work and that people like to buy, and so they make more of it. And they've hit on that combination as well. And so we buy it and we eat it and we find it rewarding. And that's where the issue is.
I think it makes perfect sense. I mean, I have people I know who are really kind of on this low-carb diet. It works for them. They can sustain it long-term. I have others who are on a low-fat diet. It works for them. They can sustain it long-term. And I think you have these examples. Herman was mentioning, the Hadza consuming large amounts of honey, right? That doesn't get any more sort of carbohydrate-y than that, right? There's just sugar, right, in that. And then you have the, some of the cultures with the best heart health, have lots of starchy tubers, and a lot of the kind of warm weather climate tribes, right? But then if you look at the Inuit or some of the tribes at the poles, they were eating these very high fat diets, not with carbohydrates at the same time, typically, right? And so I think you have these examples of the flexibility. We haven't really been exposed to those foods that contain both of those macronutrients. So this leads me to another really important topic I want to get your view on. I recently had David Ludwig.
Just before you do that, just picking up on that point, there are no tribes that you find that eat that magic composition.
Right.
Because those foods don't exist. So there isn't, there's no red oak tribe out there that are eating the composition that's, really bad for them. And I think that's a really interesting point. Sorry, I interrupted.
Right. And they're lean, and they're not obese, and they don't have a large prevalence of chronic diseases. So it makes sense. Okay, so David Ludwig, had him on recently to discuss the carbohydrate-insulin model, but this impasse in nutrition research. So we talked a lot about the kind of studies by him and Kevin Hall, some of the criticism, some of the talking back and forth. So can you give me, your perspective first on the carbohydrate-insulin model, the overall energy balance model, and where do you sit? If you think about this back and forth that has happened, there seems to be this talking past each other, and we're not really agreeing. There's criticisms of the methods or the analysis used in some of the studies. There's the crossover effects. There's the wash and wash out effects. There's the length of the design, right? There's all these questions, and I want to just get your take on where do you sit within this debate and this discussion?
So, I mean, the thing is, I think the low carb idea has got kind of mixed up with the carbohydrate insulin model. And so it's kind of been that, if people are doing low carb, they think that, okay, so I'm kind of supporting the carbohydrate insulin model. And that's led to this kind of division of, people who are doing low carb are sort of pretty, motivated to do that. And so they support the model that they think causes what they're doing. But actually, the reason why low carb works may have nothing to do with the carbohydrate insulin model. And so, it's important to kind of divorce those things in our heads before we sort of think about it. So the idea that, you take these carbs and they come into your circulation, you get a spike of insulin that packs them away, and then that can be over efficient if you have these high glycemic index carbs. And what happens is your circulating glucose then plummets. And then that acts as a stimulus to consume more food. And if it's carbohydrates that you eat, you get into this sort of yo-yo cycle of glucose going up and down and insulin going up and down to pack it away. And you get into that sort of situation where you've stored too much fat as a consequence of eating these carbs. I think it's a great idea. It's open to testing, and it's very amenable to just testing the idea. Unfortunately, most of the tests that have been done don't support it, and the tests that do support it have been done by the people who think it's a great idea. So it's not easy to then separate out what's going on. So I'll give you an example. David Ludwig did a study in some kids that was published in Pediatrics in 1999. And basically he looked for, it's like a single meal design. So basically he feeds them a meal, and then he looks for the tracks in the glucose and insulin, and he looks at how hungry they are following that, and then he looks at how much they eat at the following meal. So what he finds is that, sure enough, you get this peak in the insulin, it comes down, the glucose goes down more than what they started at, and the ones that are on the high glycemic index are more hungry and they eat more food. So, I mean, it all seems to fit. And we thought, okay, well, let's repeat that, but we'll do it in adults. So we did that a couple of years ago, and just effectively repeated the same design. There's a few little tweaks that are different. But what we found was that, okay, after they eat the high glycemic index meal, the glucose goes higher and it comes down, but it takes 5 hours before they flip over. And the flip over is very, very small at the end. And there's absolutely zero difference in how hungry they are, and there's no difference in how much food they eat. There is a difference in how much food they eat. So we use 3 diets, and there is a difference between the low and the middle diet, but no difference between the middle and the high diet in terms of the glycemic index. And so we said it doesn't support the model, because you would expect there just to be a linear relationship between, low, medium, high. It should go up in the food intake, and it should link to the hunger, which there was absolutely no effect on the hunger. And David wrote a letter to the journal saying, actually, these data do support the model because of this, the difference between the low and the medium was significant, and therefore that's it, the carbohydrate-insulin model is correct. But if you look at the totality of the data, it doesn't support the model, because there isn't an increase with the high glycemic index levels. There's zero effect on the hunger, and that flip over in the insulin is in the glucose levels is very, very small and was unrelated to how much food they ate. So most of the data, 95% of the data don't support the model, but 5% do. And so we just jumped on that 5% and said, this is it. It supports the model. So I don't think that's reasonable. I think you've got to look at all the data and then come to a decision about whether it's correct or not. And so my view was the work we've done doesn't support the model. I don't think it's correct. But I think there's a kind of broader reason why I don't believe it. And the thing is, it's got no kind of evolutionary background to it. I don't understand why we would have that system. Why would we have a system that forces us to put on more fat when we eat carbohydrates? What's the background? How did that evolve? There's no thought beyond this is a potential mechanism. Whereas the other idea that we've just spoke about, that there is this combination of fat and carbohydrate that overwhelms the homeostatic mechanism and is based on the composition of breast milk, that's an understandable story. I can get that into my head because it makes sense. There's a reason for doing that, because I just see no reason whatsoever why eating carbohydrates that are high glycemic index would lead us into an issue.
Yeah. I mean, I think this is the challenge when we have different labs producing different results and analyzing things. I mean, what you're saying makes sense to me. I just know that if David was here, who I just spoke to a couple of weeks ago, he would have an answer, right? So I think that there's
Sure.
sometimes
I mean,
there's
and
these.
don't get me
I
wrong.
think,
I mean, I think David's a very nice guy, and actually, since he wrote a paper recently in Cell Metabolism about this, we're not moving the field forward, and what we need is to break that impasse. And I've spoken to him since about that, and come up with some designs for experiments, but they're pretty expensive. And to be honest, I don't see anybody paying for those experiments to be done. But I would be delighted to work with him, and collect some data that we both agreed was well collected, and then just look at what it shows.
yeah, I think the point that I really want to highlight is that I think if you have both sort of leading experts from different labs that are involved at the start of the research to be able to have their input taken so that the design is agreed upon, and then there's no ability to say, well, but they sort of chose these things, right? They made these decisions. And I talked with a funder after I spoke with David about, can we fund more of these adversarial type of partnerships where you require to work with a group, and the donor, I won't name who it was, but they said, that's a great idea. I love that. But there's also this risk in funding, and you only want to take so many risks because if people don't get along or it doesn't work well together, that can be a loss of the funding, right? But his point was sort of, that is needed. Like, we do need donors to invest in that type of work. It's just like you said, it's not cheap. So, okay. So it's good to know. I mean, I think hopefully we can get to a place where we start doing a bit more of these kind of collaborative type of studies across labs, because I think the nutrition community and the public health in general suffers if we're not reaching consensus, right? And I think that consensus, it's okay to have differences in opinions, but the main thing that David was highlighting in that piece was, let's stop the ad hominem attacks, right? The personal attacks, the nastiness, which happens a lot online. But I think when we have these types of conversations, and I just had a conversation with Christopher Gardner, who was, I was on the recent guidelines group that worked on the new guidelines. He was on the previous committee. We talked for 3 hours on a podcast and agree about 90% of things, right? So I think there's just so much that I think can happen from respectful dialogue and discussion that I hope to see going forward,
I mean, I think you're absolutely right that, the online environment really sort of fosters that type of personal attack. I mean, it was pretty interesting that Novo Nordisk invited a lot of people who could be considered adversaries. So like David was there, Gary Taubes was there, Mark Friedman, and then myself, Kevin Hall, a couple of other people on the other side of the table. And it was really funny because on the first day, they sort of made us play games together, so we had to get out of an escape room and all this sort of stuff. And I think it was because they thought we would actually, continue that kind of online thing when we're actually facing each other, and there would be fisticuffs, and when we would be, actually adversarial in real life, and I think was completely unnecessary because of course everybody was completely charming and very nice to each other, and I thought, well, one interesting thing was right at the end of the meeting, I was eating breakfast, and Gary Taubes was there, and he left, and as he was leaving, he sort of said, "Oh, well, it was a delight to meet you, and it's really sort of good to talk about these things and everything. But you realize this means absolutely nothing about our interactions online." And then he left. And I think that was great because, of course, it doesn't. We will continue to be completely adversarial online, even though in person we actually get on really, really well.
Oh, I know. It's like the online, the whole dynamic, the algorithm, it's just designed in a way that I think it facilitates the polarization more than it exists in real life, I think. And also when you have a person in front of you, they're humanized, right? It's a different experience. Right. So, okay. So one other thing, so there's this big issue in nutrition science where we don't really know what people eat. First of all, we have very little data on what people eat. But then there's this issue of, well, what are people reporting when you do these recalls? And how do they remember? 2, are they going to be honest about it? And then overall calories, are you getting accurate assessment of what people eat? And you've done a lot of research looking at how does that compare to this doubly labeled water data? And can you adjust for those? Can you tell us a little bit about how big of the errors are there in terms of what people report and what they actually consume? And then how do you adjust for that? Is there any way to adjust for that based on this doubly labeled water data?
Yeah. So, there's lots and lots of issues with these reporting methods. And so they're not just memory issues because some of them involve people, they write down while they're eating. But, the process of writing down what you're eating may inhibit what you actually eat. And so your behavior may change just because of the recording method that you're actually utilizing at the moment. So there's lots and lots of problems. What the double labeled water technique allows us to do is to sort of say, we know how much energy you're expending. If you're in energy balance, we know you must be eating that amount of energy. So what we can do is we can calculate the amount of energy that people say they eat and compare it to the amount of energy that we know they have to eat. And then that gives us an answer to what the shortfall is. So there's a small caveat there, of course, because food intake varies day to day. And so it may just be there's a day that they didn't eat much. And so they may be completely faithfully recording what they eat, but it's just a day that they didn't eat much. At the end of the day, that doesn't matter because if it's a day that they didn't eat much, it's not a representative day of their whole life because they have to, at some point, have a day when they eat much more until it brings them back up to the average. So I don't think that's a sort of valid criticism of this approach of saying, how do these two things compare? So it turns out that there's about 30% of the energy is missing. So what that means is that some people are okay. I mean, some people sit in a zone where we can't tell the difference between what they report and what we expect. But for large numbers of people, they're just not reporting what they eat. And so you could then say, well, okay, if we know it's 30% short, we could multiply up. So if I say I eat, like 3 apples and 4 potatoes and, a piece of steak that weighs 200 grams, we could just multiply all those by 30%, and that would give us the true intake. But of course, that assumes that the content of the stuff that's not reported is exactly the same as the stuff that is reported. That seems extremely unlikely. And so I think we can quantify the size of the difference, but I don't think we can just simply say, okay, let's multiply everything up and that'll make everything okay again. And I think... Go on, carry on.
People want to report healthier foods in general. So there's this social desirability bias as well, I think, right? Like we don't want to admit the unhealthy foods that we ate, right? And that can be part of that, where you may know that 30% of it is missing, but the allocation of which 30% is hard to know.
Yeah, I mean, of course, we don't know what they're not reporting. So that's a kind of assumption that they're not reporting the unhealthy stuff. And I think it's possible, but we can't measure that. So I don't think it's possible to really say exactly what's going on. I'm really in favor of moving towards some much less invasive methods of trying to evaluate what people are eating. So for example, if you think about fatty acids in our diet, fatty acids get involved in incorporated into cell membranes. And so you could take a blood sample from somebody and look at the membranes on the red blood cells, and that would give you a really good idea of what the fatty acid composition of the diet had been. So if you then compared that to what people were reporting, you could get an idea of what they're reporting or what they're not reporting by comparing it to the blood sample. And so that similar kinds of techniques. I mean, there are also, for example, if we look at stool samples, we can look at carbon-13 levels in stool samples, and that would give us a good clue of what was coming in at the other end. So I think there are methods that we could develop that would make this field a whole lot stronger than what it is at the moment. But at the moment,
I think that's right. Biomarkers like you're talking about that can actually get a better, more accurate picture of what people are eating. I've also seen some studies that look at in the US, the misreporting, the underreporting of calories actually increased with higher BMI, with higher body mass
we
index.
find that in
So I
our
think,
data as well.
okay, so I think that's another issue that we kind of see. Okay, so there's this
So that's a
there's
big
this
problem,
issue.
of course, because what happens is,
Sure.
if you go back to the 1990s, when people were looking at these diet records, what they found is that people that are obese, or living with obesity, sorry, I should say, they were reporting lower food intake than people that were lean. And so the whole idea came about that it can't be anything to do with the food intake. It's got to be something to do with the energy expenditure. And then all that anecdotal evidence came out of the woodwork about declines in energy expenditure, which we'd spoken about already. So I think it is a problem, this field. And there are big issues when you start to associate what people say they eat and disease outcomes. So that then leads people, people make dietary advice on this, and I think it's super unsound. I don't think it's very safe at all.
It's very uncertain. I think it's there's a false sense of confidence that is placed in it by certain researchers and certain public health authorities, where really I think we need to be a bit more humble about what we know and what we don't know, and we're just kind of exploring it. It's kind of indicative. We don't really know. I think there's really another area you've sort of worked on where you have an alternative proposal. Is there's this kind of longstanding theory of this thrifty gene hypothesis, which is this idea that we evolved genes for efficient fat storage to survive famines. There's other researchers who look at if the infant was sort of malnourished when they were younger, they're potentially going to gain weight later. So there's this idea that we have always evolved in this, typically over the course of our evolution for hundreds of thousands of years, where we have an issue of not getting enough. We don't usually have the issue of we have today where we have too much. You have sort of pushed back on this idea. You came up with something. You coined a term, Drifty gene hypothesis, which is a bit different. So tell us about that in your view.
Yeah. So the Thrifty gene idea is basically this cycle of famine that is presumed to have happened in our past. So the kind of idea is that, in the past, we were always on the edge of making a balance, and we frequently went into a sort of famine situation. And during those famines, the people who previously stored fat were the ones who were going to survive because they had a buffer of energy to get them through the famine period. And then the genes that allow them to do that get selected. And so, you then get to modern society, and the people who have those genes then put on weight because they're preparing for a famine that never comes, and the result is an obesity epidemic. So I think there are several kind of levels of which that argument is just not correct. So the first sort of problem is this idea that we're always living on the edge, and there's no evidence for that at all. And you could say, well, okay, how would we ever get any evidence about, what the situation was 100,000 years ago? What are we going to do? So at the same time that all the nutritionists are saying that we don't, there wasn't enough energy, there's another group of biologists that are interested in extinctions of large animals. And so basically their idea of what it was like in the past is that you have these rampant humans going around that are killing all the large megafauna. And the numbers, the numbers that are killed, the number of animals that are made extinct is completely too many for these people to eat. So what happens is they have a thing called the overkill hypothesis. And the idea there is that early humans were so rampageous that they went around killing everything that they saw, and they were overwhelmed by food because they had so many of these big animals. So if you take an example, there are like tens of species of elephants that have been made extinct by rampaging humans. There are lots and lots of large herbivores, mammoths. Wherever you go in the world, humans have made these animals extinct, but there's not enough humans to eat all the animals that they killed. And so actually, there are these two sort of different fields. There's a nutrition field who are saying, oh, we're always living on the edge and we don't have enough food. And then there are the paleontologists who are saying, no, that's not true. Actually, we have way too much energy to eat. And so I think the whole basis of the nutrition story is a little shaky. So the second aspect of that is there's also, so we probably had lots and lots of food until we developed agriculture. So it seems likely that famine is not a thing prior to about 10,000 years ago. 10,000 years ago, we invented agriculture, and then we start to see famines because you get crop failures. And then, that's kind of issue because the food's gone for large areas and we've eaten all the animals. So there's no animals to turn to it. And so it could be that famine's a feature. But the problem with that is it's too late. It's only 10,000 years, not enough time to have all these genes and all this selection of all these genes. So the other aspect about famines that we know about that happen in the agricultural period is if the thrifty gene idea is correct, the people who die in famine should be lean people and the people who survive should be fat. And there's no evidence for that whatsoever. If you look at who dies in a famine, generally in a famine, it's not the situation that there's no food. So there's some food. There's always some food around. And the people who live are the people who can dominate that food. And so what happens is the people who survive are the people aged between about 16 and 30. The old people die because they can't compete for the food. And the young people die because they can't compete for the food. And so there are these horrible stories from famines about kids being killed and eaten and stuff like that. But basically, if you're a kid in a famine, it's not good. And if you're an old person in a famine, it's not good. But if you're in that sort of range from 16 to 30, it's fine. Generally, those are the ones that survive and get through. And there's no evidence at all that lean people died in famine situations. So the fundamental basis of the thrifty gene thing is just not there. There's no evidence that famines have been important for a long enough period. And when you do have famines, there's no evidence that the people who die in the famines are the ones that don't store fat. So it's just not a particularly great idea. If there has been enormous selection on these genes that have happened since the last 10,000 years or so, it's possible to do some analysis to detect selection of those genes in the genome. And the way that happens is because when strong selection happens, there isn't enough time to sort of break up the chunks of DNA that are being inherited. And so you get a correlation between adjacent nucleotide polymorphisms along the genome. And so you can look for that signature in connection with genes that are linked to obesity. So if you look at something like lactase, for example, which we know is something that's happened since domestication of cattle, persistence of the lactase gene, that's very strong. It's a very strong signal there. But if you look at all the obesity genes, there's nothing. There's no indication of any strong selection on those. So it seems that, the whole idea of the thrifty gene is just not there. It's just not a good idea at all. So the question is what's going on there? So this then ties into another sort of idea that's floating around in the sort of nutrition world, and that is that the fat in our bodies is regulated. So there is an idea that we have this kind of set point in our bodies and that we regulate our fat levels around this. And people sort of say like, okay, I went on a diet and I lost weight and I went back up to where I was before. And so that's evidence that there's a set point. But it's not particularly good evidence, but that's what people kind of have used. our experience of the obesity phenomenon is actually not that it's a very strongly regulated phenomenon. And so what happens is, if you think about what happens in most people is they put a bit of weight on every year and they just get a little bit fatter and a little bit fatter through time. And there's no really strong countervailing force that brings them back to this original level. So the idea that there's some sort of set point doesn't seem to match our general experience that, there is an obesity epidemic. And so I was kind of thinking about how can we reconcile these kind of ideas, and came up with this idea that, okay, maybe there's not a set point. Maybe there's actually 2 points. So maybe there's a lower point. And what happens is when we lose a lot of weight, there is a resistive force to sort of push us back up. And so there's like a lower intervention. And if you go below that lower intervention point, there is a physiological reaction. And then there's a gap where nothing happens, a sort of zone of indifference with our body weight. And then at some point, there's an upper intervention point that stops us getting fatter. So some people are quite lucky, and these two things are quite close together. And so they're the people who don't really put on weight, because what happens is when they go up, they hit their upper intervention point and they get some reaction and it stops them putting on weight. Whereas other people have a really high intervention point and they just grow and grow and grow and grow, and they never have any reaction against that. So that explains the sort of obesity epidemic. And I was really pleased with the idea, and I thought, oh, that's really good. And then I found out somebody else had had the same idea like 10 years before. So it's, there's no new ideas in biology. The point about this dual intervention thing is, why does that happen? Why would you get a situation where this upper intervention point was different in different people? And so you have to think about why those points exist in the first place. And so what I suggested is that we need a certain amount of fat as a buffer against periodic lack of supply. Okay, so this is going to sound like the Thrifty gene hypothesis again, because we're saying it's used to get you through periods when there's no energy. But the difference here is, what I'm suggesting is that those periods when there's no food is actually when you get sick. So if you get an infection or you get ill for some reason, or you break your leg or something like that, there are very strong mechanisms in your brain that shut off you feeling hungry. So it's post-infection anorexia, very well established phenomenon. It's very well characterized what's going on in the brain. And so you need a buffer to get you through those periods. So there can be abundant food absolutely everywhere, but if you get ill, you need a buffer to get you through that period of illness. And it turns out that, if you think about a normal BMI, normal person, normal BMI, we store about enough energy to get us through about 2 to 3 weeks. And that's probably about the duration of a typical infection. So the levels that normal BMI represent kind of match into that idea. And that's been around forever, so that's presages agriculture development and stuff like that. So it's nothing to do with famines and thrifty genes. It's all about it. So what about the upper intervention point? Why do we have an upper intervention point? Because, if we stored a bit more fat, it would get us through a longer period of danger. And the problem with storing more fat is it increases your risk of predation. And so if you're trying to run around and the environment's got lots of predators in it, then you may be more at risk if you're carrying around more fat. And in fact, if you look back to, 4 million years ago, 3 million years ago, Australopithecus, Australopithecus, they seem to have been preyed on by a particular clade of carnivores that no longer exists. So dinofelids were specialist predators of Australopithecines. And so predation risk was a really serious thing for these animals, pre-human animals. And so they probably had a very tight regulation on their body weight so that they didn't put on too much weight. 2 million years ago, what happens? Homo erectus evolves, social animal, invents fire, invents weapons, starts to extinct all these mega herbivores, but also starts to kill all the predators. And so then the risk of predation falls through the floor. There's nothing going on. So what would happen in that situation? So the thing is, there's like an absence of selection now. So we have that system in our brains, but it's not doing anything. So nothing would really happen. But slowly there would be mutations and it would start to fall apart. But it wouldn't fall apart in everybody. So what happens then is you get a diversity of genes that influence this upper intervention point. And they're not under any sort of selection. They're just drifting around through time. And so because
they're not, there's no consequence of having that excess body fatness, right? No major consequence.
Right. So, but I mean, there's no drive to go up. It's just the point at which you stop has changed. So there's no disadvantage to it, and it's just, it's drifting around, but there's no consequences until you get into an environment where there's lots of food that are pushing you upwards. And then
lots of food with carbohydrates, with fats, combined with packaging, with marketing, with food markets around us. So
once you get in that environment, you go up to this, to your personal upper intervention point. And some people have a higher point than other people. And so I called it the Drifty gene hypothesis to sort of counterbalance the Thrifty gene hypothesis, because these genes are drifting away.
Yeah. And I think it makes a lot of sense, and it explains why today we have, like you mentioned, we have these people who, no matter what they do, they just don't seem to gain too much weight, and other people who do the same thing and gain a lot of weight, right? It's not really a fair thing, but those are those genetics passing, that distributing, right, drifting. And I think that you're saying there's not that selective pressure of predation on people where you really had to stay kind of in the normal range to not get eaten. I've taken up a lot of your time, but I wanted to ask one more question, because you've been sort of a leader on very different topics. Like we've talked about the energy balance model. We've talked about doubly labeled water. We've talked about this Drifty gene hypothesis. And there's one other topic that gets brought up around this, the sort of amount of calories and energy expenditure. And it's sort of calorie restriction, energy restriction for longevity. And there's this kind of idea out there that cutting calories has mostly just positive benefits for longevity. And you've pushed back on that with another term. I want to get this right. The clean cupboards idea. So tell us about this as we wrap up. Tell us about what is this concept, because it's not just positive outcomes that you're noticing or you've detected from this research, right? It's more a bit of a trade-off in this calorie restriction.
I mean, it's pretty clear calorie restriction does have lots of benefits in terms of lifespan and health span. And so the question that I've been involved in is why? Why does it has that? Because it's not completely intuitively obvious. And the classic explanation that was thought up in the 1970s by a guy called Tom Kirkwood is called the disposable soma hypothesis. And so the disposable soma hypothesis is basically that we take in energy and we can use energy for two things. We can either use it for reproduction, so we can put energy into making more babies, or we can use it to protect our bodies and protect our soma. And what he claims is that the reason that we age and die is principally because of reproduction, that we put energy into reproduction and we don't put enough into our soma and we don't protect it. And the idea of the sort of using the disposable soma to explain calorie restriction is that if you reduce the total, that what you do is you kind of decide, okay, well, I'm going to switch off reproduction. And because the reproduction is more than the reduction, you then have a little surplus, and that surplus you can stick into somatic maintenance, and then you end up that you live longer. So I was pretty interested in that idea because we've done lots of studies of caloric restriction in mice, and there are two sort of fundamental aspects of doing caloric restriction in mice that just don't fit that model. So the first one is they're not reproducing. So from the start, the amount of energy that they're eating is not going to reproduction. It's only going to somatic maintenance. And if you cut that energy, the more you cut it to a certain point, the better it is, the longer you live. So if you have 10% less, you live a certain amount longer. But if you have 20% less, you live even longer. So it's the opposite of what you predict from the disposable soma hypothesis. And so I was kind of thinking about, how that comes about and what happens. And the thing about the disposable soma is it's kind of like the animal has a strategy in mind. So what it's trying to do is it's trying to survive this period of food shortage so that it can get through and reproduce again. That's the kind of idea that's there. And I was thinking that actually the animals that we were studying, they don't have that sense of the future. So they're not trying to survive till some mythical future date when they can reproduce again. What they're trying to do is just get to tomorrow. So they have an immediate problem that we've not given them enough calories, and they have to deal with that now. And all they're trying to do is just survive now until the next set of food comes the next day. And so how did they do that? Well, so the clean cupboards idea was a kind of attempt to explain what's going on. So let's imagine that, and actually I came up with this idea just before COVID, and it was published in 2020, and right in the middle of the COVID crisis. And lots of people wrote to me and said, "Oh, that's a great analogy. I was, in COVID and everything as well." But this actually was before COVID, and I it just happened to work. So let's imagine you're locked down for some reason. You're locked in your apartment, and every day somebody comes to your apartment with your food. So they give you the amount of food that you need to eat, and it's the amount that you need, and you eat it and everything's fine. But then one day, the amount of food they bring is less than what you actually got in the day before. So it's now 10% less. So what do? You have an immediate problem. So probably the first day you wouldn't do anything and you'd burn a bit of your body fat and you'd start to lose a bit of weight. But then after a while, that wouldn't be enough, so you need to do something else. So if you're anybody like me, you've probably got a kitchen full of cupboards that have got food that you bought that you didn't need. You would start to eat that. So you would start to work your way through this pantry and cupboards and your fridge and everything you would eat. And so slowly over time, you would get a spectacularly clean set of cupboards in your house. So there's no strategy. You weren't aiming to do that, but it would be there. You would have this beautiful set of cupboards. But then, once you'd cleaned all the cupboards, you would have nothing else to eat. So you might start to eat the houseplant, because you still need to eat something. You still need to do something. And so you have this kind of situation where you have some really nice things that have resulted from the lack of food, but you have some things that are not so great. But they all came about because what you're trying to do is just make a balance. You're just trying to get to the next food delivery the next day. And so the analogy works in the mouse and presumably in humans as well under calorie restriction, is that what happens is when there's a shortage of the food, first thing you do is you start to run down your fat stores. So we know in the mouse, they run down their fat stores over the first 30 days of restriction, and then they don't drop them any lower. And then what they start to do is they start to clean the cupboard. So in your cells in your body, you have lots of junk protein, and it's just kind of hanging about. It's not really doing anything particularly bad, but it's not doing anything good either. But if it accumulates, it can be bad. And so you start to break down all those junk proteins, and then you start to break down the dead cells that are in your body. And so you clean up your body. And the benefits of that are that you live longer. So it's just a kind of, it's no strategy involved, but it has those benefits. But it can go a bit too far, like eating the houseplants. And so you get some negative things that happen as well. So for example, calorie-restricted animals are really not so great at immune function. So if you give them exposure to a pathogen, they find it really hard to fight it off. And if you cut them, they find it really hard to fix the wounds and everything. So there are downsides as well as upsides. And that's a difference to disposable soma, because disposable soma says there's only upsides, because all you're doing is investing the energy to live longer. Whereas in the clean cupboards, you get both upsides and downsides. And on balance are good, but there are bad things that are happening as well, because you're only doing it to get to tomorrow.
Yeah. Yeah, it makes sense. And I think one of those downsides also is you don't want to lose too much lean mass as you're aging, right? There's falls, infections are one way people die, but falls, broken bones, ending up in the hospital, those are other ways that people die. So there is this kind of balance, and I appreciate that perspective on it. Dr. John Speakman, I thank you so much for joining me. It's been a pleasure.
Thanks for interviewing me. It's a real pleasure to share this stuff. And I've seen another couple of your interviews, so it's a super interesting podcast, and I'm very honored to be on it. Thank you very much.